My name is Yuki Yoshida from Keio University, and I'd like to talk about my work on tardigrades, and they're pronounced tardigrades. And they're very beautiful creatures that looks like this. And first of all, I believe that most of the people here do not know tardigrades. They're animals with body length about 0.1 to 1 millimeters, and this is a real movie about them. And they live in a variety of environments. And one interesting thing about these creatures is that when the surrounding environment around them desiccates, dries out, they go into a state called like a ton. They go into a state called anhydrobiosis, also known as a ton. And when they regain water again, they revive animal activities like this. They're getting water right now, and their right leg around here starts moving in about a second or two. They start moving all their legs and start moving all away. And interestingly, in this anhydrobiotic state, the ton state, they are capable of tolerating various extreme environments, such as 100 degrees, UVC, gamma ray, and a vacuum space. And in 2015, a group led by Bob Goldstein from University of North Carolina reported a genome of Hypsibius dujardini, a freshwater tardigrade. And interestingly, they reported that they have found an abnormally high ratio of horizontal gene transfers, HZT from now on, which totaled it to 17.5%. And some of you may have seen it in Twitter with the hashtag Tardigate. And a lot of groups have researched this genome, along with including our group, and independently sequenced this Hypsibius dujardini, and have not been able to find this abnormal high ratio. And other groups have found heavy bacterial contamination, which totals up to about 25%, and 3 nearly complete bacterial genomes in this genome assembly. And so to fix this problem, we would need a contamination-free genome, which I don't get the meaning of both, but a contamination-free genome. And in 2016, a group led by Kunieda Takekazu in Japan, Tokyo University, has sequenced the genome of Ramazzottius varieornatus, which is a very, very close tardigrade in the same Hypsibidae family. And this group has previously identified several protection genes, which is specific only in tardigrades. And these genes localizes in various cellular compartments, such as the cytoplasm, the mitochondria, and the nucleus. And interestingly, this gene called DSUP is capable of getting human cells infected with this DSUP has higher X-ray tolerance, which suggests that this gene is related to cellular protection in some way. And the genome of Ramazzottius analysis suggested that there's severe loss in stress response signaling pathways, such as this HIF1A, which is found to be mutated in cancer cells. And these findings suggest the fundamentals of the molecular mechanisms of anhydrobiosis. However, these only have been seen in Ramazzottius, only in Ramazzottius, so we require to see them in other anhydrobiotic capable tardigrades. So in this study, we have resequenced and assembled the genome of Hypsibius dujardini, where previous assemblies had bacterial contaminations and fragmentations due to the fact that they've mostly used only short reads in most studies, and uncollapsed heterozygosity, which resulted in an increase of the genome size, more succeeding than the estimated genome size from, for example, flow cytometry experiments. And we have used short reads from Illuminus and a long read using PacBio technology. And by using a heterozygosity-aware assembly system and RNA-seq data from various life stages, we obtained a high-quality genome and a high-quality gene prediction gene set. And using this high-quality genome, we tackled these 3 problems: the horizontal gene transfers and anhydrobiosis mechanisms, and the phylogeny of tardigrade, the phylum Tardigrade. And first of all, the method of the assembly. First, the genome of Hypsibius dujardini is about 100 megabases, and previous assemblies had about 130 megabases. So we need to fix this heterozygosity problem. And first of all, we have employed genome sequencing from a single individual, which is about 500 picograms, so it's going to be a ultra-low input DNA sequencing. And we have already reported this method in a paper in about 1 year ago. And using this method, we are capable of minimizing the contamination because we are physically looking at the tardigrade and adding antibiotics to minimize this contamination. And using this data, we excluded non-Hypsibius DNA seq reads from the data from the UNC and assembled this data with using the Platanus assembler, which is a heterozygosity aware assembler. And for the PacBio reads, which has about a 10.6 N50 length, we assembled this long reads with using the Falcon assembler. And by using this Falcon assembly to extend this Platanus assembler with PB jelly and S-based long reads and remove low quality contings such as those that are very short or ones with low coverage. And after error correction of the pylon, we obtained the final assembly, which is 104 megabases, which is very, very close to the size, genome size estimated from previous flow cytometry experiments. And our genome has far higher genome metrics compared to previous Hypsibius assemblies with N50 length about 342 kilobases, longest scaffold at 2.1 megabases. And the genome Ramazzottius, the one reported in 2016, has similar high metrics. And after using various RNA-seq data of the embryo, juvenile, active, and ton states, and using breaker version 1.9, we obtained about 20,000 genes and 14,000 genes in each tardigrade with high Busco scores. And the first thing we did with using this high-quality genome was to figure out how much horizontal gene transfers there were. And after using the method proposed by Boschetti in 2013, we found around 1% of the whole genome to be true horizontal gene transfers, which is, not that much high because C. elegans has about 1.2% and Drosophila has about 0.5. And for Hypsibius, we have validated these horizontal gene candidates using phylogenetic and conservation between tardigrades. So what we think is that tardigrades don't have that much horizontal gene transfers. And to add to this, we have also calculated the horizontal gene transfer ratios in various excitozoans. And the red is the ones that are the genomes that are submitted to Ensemble, and the blue ones is pretty much interesting, but we have re-annotated these genomes using Augustus. And again, calculate the HGT ratios in each of the genomes, and found that in arthropods, a high level horizontal gene ratios are going up to about 15% in this, and Drosophila has about 5%. So what we think is that the algorithm used in these gene prediction softwares obviously have several false positive gene predictions, and these predictions may be detected as horizontal gene transfers. And for next, I want to talk about the mechanisms in anhydrobiosis. And the two tardigrades that I have explained, Ramazzottius, the brown one, and Hypsibius, the white one, live in two completely different habitats, which Ramazzottius lives on a moss found on bridge, and Hypsibius lives in a pond. So Ramazzottius is more prone to desiccation compared to Hypsibius. And Ramazzottius, the brown one, is capable of rapid desiccation, which takes about less than 30 minutes, while Hypsibius requires at least 24, and it takes up to 24 to about 48 hours. So one thing interesting about them is that although they are very similar, they're very phylogenetically close, but they have completely different anhydrobiosis mechanisms. And previous studies have found that the Hypsibius requires de novo transcription, new transcription, to enable a successful anhydrobiosis. So what we propose is that anhydrobiosis-related genes, such as the genes I've shown in about the 5th slide, the ones about the protection, may be highly induced only in Hypsibius. So what we did is the RNA-seq analysis of the active stage and ton stage of slowly and rapidly desiccated Ramazzottius and normally desiccated Hypsibius. And what we have found is that this plot shows the expression in ton state and fold change of active and ton state in each tardigrade. And these are all differentially expressed genes. And the number of differentially expressed genes is higher compared in Hypsibius compared to Ramazzottius, going up to about 7% in Hypsibius. So the differentially expressed genes number is just higher. But you can see that the induction genes are highly induced in Hypsibius compared to Ramazzottius, and this is a significant change. And so then the differentially expressed genes contains various protection-related genes, such as the COS, SAS, the ones that COS localizes in the cytoplasm, and the MOS and RV LEM that localizes into the mitochondria. So what we think is that the difference in habitat may be contributing to may be the reason for this different anhydrobiosis mechanism, which Hypsibius requires new transcription of anhydrobiosis-related genes, where Ramazzottius doesn't require induction, but has constitutively a high expression during out their life stages. And last of all, I'd like to talk about the phylogeny of this tardigrade. This is a tardigrade looking for the stomach side. And you can see that it's kind of a weird animal that has 8 legs. And previous studies from morphological and molecular evidence has completely different conclusions, where, for example, the structure of the pharynx and phylogenetics has proposed that tardigrades are more related to nematodes compared to arthropods. However, the structure of the central nervous system and microRNA existence patterns suggest a more tardigrade and plus arthropod clade compared to nematodes. So a lot of this discussion has been going on for this about 20, 30 years. And from our two tardigrade genomes, we have conducted single copy ortholog gene trees and Hox gene existence patterns for both of the tardigrades. And for the left side, you can see that the green is nematodes and red is tardigrades. And you can see that these two form a sister clade. And in Hox gene existence patterns, the red is right here, tardigrades are right here, and tardigrades show a profile very similar to nematodes compared to arthropods. So our two genomes suggest that tardigrades are more similar, well, related to nematodes compared to arthropods. And for the conclusions, we have assembled a 104 megabase heterozygous genome of Hypsibius dujardini and obtained a high-quality genome gene set. And using this gene set, we have found that, well, the 17.5% is, well, wrong, and we have found that the true horizontal gene transfer ratio is very limited to about 1%. And as for the anhydrobiosis mechanisms, we have found that the environment which they live in leads to the difference in the anhydrobiosis mechanisms, which can be explained by gene regulation. And for the phylogeny, we have provided evidence for the tardigrade and nematode clade. However, some evidence still suggests that tardigrade and arthropod clade might be there. And interestingly, during this analysis, we have identified several gene families that are very, very highly conserved from the excitosome common ancestors. In some cases, completely conserved within arthropods and tardigrades. That has completely no annotation at all. So we were surprised to find these sort of genes and these genes may be targets for the later evolutionary analysis. And this work has, well, very happily been accepted to PLOS Biology and will be released in about 2 days, I think. And please, if you like this talk, take a look at this paper. And I would like to acknowledge these members. I study at Cambridge University, and this work was in collaboration with Edinburgh University in Scotland and with Guneda Takekazu in Tokyo University. Thank you for your attention.
Thank you. So we have time for one question, but I would need to ask you to please use the microphones in the audience.
For the differential transcriptomic analysis, what tissues were used?
Since there's a very small, we have used a bulk, about 30 animals for each sample.
I see. So is that a concern? I mean, what are it could be a false signals. I mean, have you thought about it?
From our present findings, we do not think it's that much of a concern. It has targets have about 1,000 cells, and well, looking at single cells might show some kind of change, but well, looking at the bulk itself will prove to be right, I think.
All right, so let's